The process of meiosis is one that generates genetic variation through recombination, independent assortment, and random segregation. Meiosis produces 4 haploid daughter cells that are different from the parent and sibling cells. In humans, the haploid cells made in meiosis are sperm and eggs. When they join in fertilization, the 2 haploid sets of chromosomes form a complete diploid set, a new genome. During meiosis, one gamete from each parent combines to form a zygote. My inquiry question based on meiosis is how significant is crossing over in genetic variation? Before entering meiosis 1, a cell must first go through interphase where all chromosomes are duplicated in a process similar to chromosome replication prior to mitosis. During prophase 1, the chromosomes begin to condense and pair up, becoming visible under a microscope. Each chromosome carefully aligns with its homologous partner so that the two match up at corresponding positions along their full length. By metaphase 1, the nuclear membrane has disappeared and the microtubules form spindles. Spindle fibers attach to only one side of each centromere, and the 2 homologous chromosomes attach to the microtubules, orienting from opposite poles. Each pair of chromosomes then lines up on the metaphase plate. The orientation of each pair on the spindle axis is random. Either the paternal or maternal homologue may orient towards a given pole. Each orientation produces gametes with different combinations of parental chromosomes. Random segregation is when the 2 chromosomes in meiosis are separated randomly to the reproductive cell's gametes. There is no way of telling which chromosome will end up in each gamete. As a result of segregation, each diploid parent passes a random allele for each trait to his or her offspring during fertilization. Crossing over during meiosis allows recombination of genes between homologous pairs. This alters the linkage between genes on the same chromosome. Crossing over occurs in the region between the 2 genes and is only between a homologous pair. The possible haploid cells after meiosis have 4 genetic combinations. The model shows that the 2 will come from chromatids that did not participate crossing over and thus will still have the parental combination of alleles, and the other 2 will be recombinant. However, both chromatids participate in crossing over. The model is limited here as it doesn't show this. Crossing over creates new combinations of genes in the gametes that are not found in either parent, contributing significantly to genetic variation. Independent assortment states that genes are inherited independently of one another. That is, the formation of random combinations of chromosomes as the lining up of one set of chromosomes does not influence the lining up of chromosomes above nor below that pair of chromosomes. This leads to variation as this process limits duplication of the inheritance of similar maternal and paternal chromosomes and hence genes. Imagine an individual has 2 different alleles, versions of a gene for eye color, blue and brown, and 2 different alleles for hair color, blonde and black. If a gamete produced by the individual contains the blue eye color allele, it has an equally good chance of containing the blonde or black hair color allele. Chromosomes segregate during meiosis 1, and during prophase 1, homologous chromosomes pair up and become attached to one another. At this time, crossing over moves sections of DNA between homologous chromosomes and allows for independent assortment. In anaphase 1, the microtubules of the spindle fibers shorten and pull chromosomes toward the poles, taking both sister chromatids with them. Each pole ends up with a complete haploid set of chromosomes consisting of one member of the homologous pair. During telophase 1, the nuclear membrane reforms around the daughter cell. Each daughter nucleus contains two sister chromatids for each chromosome attached to a common centromere. Because of crossing over, the 2 sister chromatids are not identical. Cytokinesis is the process of cell division which divides the cytoplasm of the parental cell into 2 daughter cells. During prophase 2, the nuclear envelope breaks down and new spindle fibers form. In metaphase 2, spindle fibers bind to both sides of the centromere. During anaphase 2, the spindle fibers contract and the sister chromatids move towards opposite poles. In telophase 2, nuclear envelopes reform around the sets of daughter chromosomes. Cytokinesis occurs again to divide the cytoplasm of the parental cell into 4 daughter cells.